All right, hello. How are you guys? Hope you did good on your chemistry this morning. Thank you for joining me with the review. Let's get right to it. So, last Friday we already last Thursday we already took up part of the review sheet. So I'm just gonna continue from where we left off. If along the way you have any questions, like any additional concepts you want me to go through, or things that you don't understand, again, this is just like a regular class, okay? You just interrupt me and ask, just like you would in the classroom. So here we go. This is the review package, the green package, and this is about lecture 7. Here, a DNA segment has 300 nucleotides. First of all, like I also want to tell you that if you are preparing for the test, there are a couple of things that you should be doing. Number one, you absolutely should go through every questions in the package. That's why I'm taking it up with you. Okay, no, don't even bother going through the notes and the study guide just yet. Okay, just focus on knowing the answers for all the questions on the review sheet here. Okay, that is gonna get you to a solid B. All right. And then you should review the poll questions, concept check questions that we do in class. The ones that I tell you to do 1, 2, 3, 4 in class. Those questions are also important. The answers are on Luminate as well. And then as a final brush up, then maybe review your quiz 3 and quiz 4. That alone should be enough. Okay, don't go crazy, making new notes and stuff like that. Just do the review package. Okay, concept track questions, quiz 3, quiz 4, and then if you have time, maybe do a little bit of the study guide as well. Then you should be golden for the test. Okay, this is lecture 7. A DNA segment has 300 nucleotides. Okay, 300 nucleotides. And how many amino acids does this DNA segment encode? What assumption did you make? The key thing here to know is 3 nucleotides. 3 nucleotides equal to 1 codon, and 1 codon equals to 1 amino acid. Right? So like if you have 300 nucleotides, that's going to be equivalent to, you divide by 3, which is 100 amino acid. 100 amino acids. What assumption did you make? Well, a couple of assumptions here. I'll write the assumptions in purple. Number one, no introns in the DNA. Okay, everything is expressed, everything is going to be converted into the amino acid, and you didn't cut anything out. That's an assumption that we made. Okay. If it's intron, they are non-coding region that needs to be removed during post-transcriptional modifications. Another assumption that we made would be no stop codon. Okay, if there is a stop codon somewhere in the DNA segment, then it's not going to be, as long as 100 amino acids. It's going to stop like somewhere in between. Okay, so these are the assumptions that you might have consciously made or not, right? It really depends. Okay, moving on. That was A. B, the 24th nucleotide is deleted. How many amino acid will be unaffected by the deletion? Okay, so like if it's 24th nucleotide, so like that means 24 divided by 3. What's 24 divided by 3, huh? 24 divided by 3 is 8 amino acid. Right? So these first 8 amino acids will be okay. Okay. Actually, you know what? Not 8. Okay. The 20. Sorry, let me do this again. Dot 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 dot, and then 22, 23, 24. Okay, so this would have been the eighth amino acid, because every three is going to be a codon, right? And what they're saying is we're gonna delete the 24th. So if this goes, then the eighth amino acid is actually going to be wrong. Okay? So the first seven, first to seven are okay. So if you delete the 24th, in fact, if you delete the 22nd, 23rd, or 24th, all will give you the same answer. Okay, 1 to 7 will be okay, but the eighth one will not be okay. If you delete the 25th, then first 8 would have been okay. Does that make sense? Okay. Moving on. Part C here. A nucleotide is inserted after the 50th. Inserted after the 50th nucleotide. How many will be unaffected? So again, you can try to do 50 divided by 3. Okay, 50 divided by 3 is 3 and then 26, 16.66666, right? So that means, I mean, 50 is not a multiple of 3, but 51 is. So 51, 50th, and 49. So that would be the 17th amino acid. Okay. So if you insert something after the 50th, go here, then the 17 is going to be messed up. And number 1, first to 16 would be okay. All right. So basically you have to figure out like where the triplet is, and then everything before that point would be unaffected. Okay. All right, let's do this one. So write the mRNA sequence. This is the coding strand. If we are given the coding strand, then the mRNA sequence is identical, but the T becomes the U. On the test, I will give you the table, and it's going to be last page, so you can rip it out. So you don't have to be flipping back and forth so many times to refer to it. And so let's take a look at the code here. Of course, you probably know amino acid here. Of course, you probably know the first one is MET. And then you just look up so GAC right here, which is ASP. By now, you should know how to use this, just like this is almost like the periodic table, right? But for biology. CAA is GLN, and don't forget to put stop. Okay. So we roll the mRNA sequence, we translate it into the amino acid. Okay, some of you lost marks on the quiz number 4 because I was asking you for the protein sequence. Protein sequence means amino acid, and then some of you wrote the ACTG instead. So just be careful on the final test. Right? There's lots of time, okay? You will have enough time. Just take your time, read the question carefully, see what it is asking you to do. Create a nonsense mutation by either base substitution or insertion. This question was pretty well done on the quiz, but let's just go through it again. Base substitution means I can only replace the nucleotide. I'm not putting in extra things. I'm not taking them out. So what we're going to be having is we need to change it to a stop codon. Okay, and we don't change the last one because the last one is already a stop codon, right? So there's only one place we can do it, which is right here. If I change it to T, then it's going to be a UAA, which would be a stop codon. Okay, so that's base substitution. If I want to create a nonsense mutation with an insertion, then I have to add an extra nucleotide that would result in a stop codon. So I see GA here. What I can do is I can put a T right there, and that would cause it to become stop, because then the ribosome will read it as AUG and then, UGA, right? Technically, I should put a U here because this is the mRNA, but you would have to put it in the original sequence like that. Okay. So that is that. Any questions? Okay, create a neutral mutation by base substitution. Neutral mutation means we are going to not change the sequence. So all you have to do is look up like GAC or something. So GAC is right here, which is ASP. So if I wanted to stay ASP, stay as ASP, then we change GAC to GAU. Okay, so GAU, which means that the original, it's going to be a T. So then it's going to be no change. All right. Okay, we're going to move on. Hemoglobin is an oxygen-carrying protein found in red cells. Its quaternary structure contains 2 alpha chains and 2 beta chains. Thalassemia is an inherited blood disorder that causes your body to have less hemoglobin than normal. In some forms of thalassemia, the beta chains are completely missing in some hemoglobin. What kind of mutation is the likely cause of this? Explain. So if the whole thing is missing, there's only one kind of mutation that would cause a whole chunk of proteins to be missing, right? And which one would that be? Anybody?
Nonsense.
That's right. This would be a nonsense mutation. Okay, so nonsense mutation causes premature stop. In translation, resulting in shorten or missing protein, amino acid, right? Proteins, you can say. Okay. Explain the likely outcome of these mutations. Be sure to consider where it occurs. Okay, so missense, let's talk about missense. Okay. If replaced by by amino acid of the same property with the same with the same property, e.g., like acidic to acidic, as opposed to acidic to basic, or like polar to non-polar, right? So if you missense is replacing the amino acid, right? So if you replace the amino acid with the same property at a non-vital location, okay, then minimal effect. Okay? Non-vital location means it's not like a key functioning part of the protein. In the context of an enzyme, it would be like the active site. So a non-vital location would be anywhere else other than the active site. Okay, so like replacing the amino acid, if it stays the same, even if it's in the active site, probably less harmful than replacing it with something that has a different property. Okay, so that's the first point. More severe outcomes if it happens in an active site or recognition site. Nonsense earlier in the sequence is generally worse. Okay, same thing with frame shift. Okay? Because these things, the earlier it happens, the more you gonna mess up. Okay? You're gonna lose like a whole chunk of amino acids if the nonsense happens early on. Frame shift, the earlier it happens, the more mistakes you're gonna have in your sequence. Right? Remember, everything after the point of insertion or deletion that's not a multiple of three will result in massive, extensive missense mutation. Okay, so your whole protein's basically like 80% of it is gonna be wrong. So these are worse. Okay. Basically, I'm gonna probably give you a bunch of mutations, and then I'm gonna ask you like which one will likely have the worst outcome or have least impact, right? Then you have to know how to look for it. Okay. Okay. Use a diagram to illustrate the following terms. We've done this a couple of times, but let's go through it again. This whole thing is basically a gene, and the gene has a beginning and an end, right? The beginning and an end. The beginning is called promoter, which is AT rich. Okay, there was a quiz question. What was it, like quiz number 3 or something? Let's give me a second. Right, there was bonus question in quiz number 3 that I asked you to find the promoter. So promoter is rich in A and T because it's easier to pull apart. There are only two hydrogen bonds holding the A's and the T's, as opposed to three hydrogen bonds holding the C's and the G's. So, over here, I would say this is the promoter. This is the part that has a lot of A's and T's. You know what? Let's use green. That's the promoter. Okay. And then over here, this is the end of a gene, which we call terminator. So that's going to get converted into the messenger RNA, pre-messenger mRNA. And now this thing is going to have some, coding portion, and then it's going to have some non-coding portion. Okay. The non-coding, we call them introns, which we have to remove. Whereas these ones, the blue ones, that's the exon, okay, exons, which are coding. So then afterwards, after the modification, we're going to cut out all the non-coding ones. Okay, put a 5 prime, 3 prime here. And you're going to put a special G on the 5 prime. That's called a G cap. And then you're going to put a bunch of A here on the other end. Okay, so that's the poly A tail. And that's the G cap. Okay, and that would be the finished mRNA. Okay, so that's the process of transcription, basically. Okay, explain the relationship between codon, anti-codon, tRNA, and amino acid. Basically, this is the mRNA, and then you're gonna have the ribosome. Okay, I'm not gonna ask you to explain this on the test, okay? But you have to know the relationship in order to answer the questions. Okay. So, on the messenger RNA, you're gonna have the codon. Okay, UUU, CGA, AUC, whatever, right? And the key thing to know is you're gonna have the transfer RNA, which looks like a clover leaf, and it's gonna be carrying an amino acid. Okay, so this one is gonna be PHE. And how does the transfer RNA know it's the right amino acid? Well, at the bottom of the transfer RNA, there is going to be the anti-codon, which would be complementary to the codon. Okay, and this thing here is tRNA. Okay, so if the anti-codon and the codon matches up, complementary, then it will stay and it will leave behind the amino acid. Okay. Remember, do not look up the anti-codon on this table. Okay, this table is only for codon. If I give you an anti-codon and I ask you what amino acid it corresponds to, you have to change the anti-codon to the codon first. Okay, so for example, if I say the anti-codon on a tRNA molecule is ACC. Okay. What amino acid does it carry? And what amino acid does it carry? Okay, so let's take a look. So this is, of course, the anti-codon, and we're not going to look it up. Instead, we are going to look up. What are we going to look up?
We're gonna hook up at codon,
That's right, which is what?
which is UGG.
UGG, that's right. Does everybody understand why it is UGG? Okay, if you don't understand, you should ask. You need to know how to do this on the test. So A pairs with U, C pairs with G, right? So UGG, where is UGG? UGG is TRP. Yeah, so like you would have the tRNA, it would be carrying the TRP, and then down here would be ACs. Okay, that is all. Okay, that's it for lecture 7. We'll move on to lecture 8 now. Heredity. Okay, so obviously expect uh, to be asked to solve some of the genetic problems, okay, like the ones we did in lecture 4. It's sorry, in quiz number 4. It's not going to be more difficult than that. Okay, the only thing that you haven't been quizzed on is the sex-linked stuff. So maybe we'll do some practice with those. Okay, here we go. Explain the significance of crossing over. How, why, where does it happen? So crossing over is the happening in prophase 1 of meiosis, and it's the process where the homologous chromosomes will come together and form a tetrad. Okay, so this is tetrad, and these are homologous chromosomes. Okay, and that's like the crossing over here, right? Which is the exchange of genetic information to create genetic variation in gametes. Okay. Where? In the cytoplasm. Okay. When? Prophase 1. Okay. How is like forming the tetrad? Why is to create genetic variation. Okay. So afterwards, you're gonna have a bit of the red on the blue and a bit of the blue on the red. You can draw that if you want, but I'm sure you remember it from class, right? What is the purpose of meiosis? Sexual reproduction. Okay. Create gametes for sexual reproduction. Where does it happen? Gonads, right? Which is the testes and ovaries. Result? Sperm and eggs, okay, which are haploid cells. Okay, so remember, the sperm is N plus the egg, which is N, and then you get 2N. We call that the zygote. Okay? What is non-disjunction? Okay, I'll show you the question, all right? Free question. There you go. All right, hold on. That's a question you have to know. Okay? Yes, Annie, you have a question or
Yeah.
yeah, go ahead.
Could you just scroll up like
There you go.
3 lines quick?
There you go.
Thank you.
Yeah. I'm gonna launch the poll. All right, you guys can figure it out. Non-disjunction. Just do this question right here. Come on, everybody choose something. I only have 5 answers. That's less than what I get in class. What is non-disjunction? Okay, that's fine. The correct answer, non-disjunction, is failure to separate chromosomes in anaphase 1 or anaphase 2. Okay, if you see non-disjunction on the test, choose C. Okay, there you go. Free question. Everybody get 1 mark. Okay, that's non-disjunction. They don't separate, and as a result, you get extra chromosome in some gametes and missing chromosomes in others. Okay, next. Practice all different types of genetic problems. Okay, this one I cannot help you. Okay, like you have to know how to do them. Okay, the monohybrid is one trait, dihybrid. Just do quiz 4 again, especially if you didn't do well on quiz 4. Okay, watch the review video again and then try to get the right answer. Okay, and do corrections on your quiz. I'll do a sex link with you though. Okay, so let's say we have a colorblind woman. Okay, marry a normal vision man. Okay, probably you should say man with normal vision. Okay, so we're gonna try to figure out uh the outcomes. So color blindness is an X-linked recessive condition. That means the woman who is colorblind is going to be XCXC. Both of her X chromosomes will have the colorblind gene. Okay. And for the man with normal vision, it's just XY. We do not put the C on this person because he doesn't have color blindness. Okay. So then we go ahead and do the Punnett square. I'm going to put these gametes here, XC, XC. I'm going to put the man up here, XY. Then we have XXC and XXC again. XCY, XCY. Okay, so we will have 50% female, 50% normal vision. Okay, girls. And then you will have 50% colorblind son. Okay, just do it as an overall. Okay, do it for all four. It's probably easier. There's some confusion about separating into boys and girls. Don't worry about it, okay? Just count them as a whole. Okay, that means 50% of the kids is going to be like a girl with normal vision. 50% chance you're going to get sons who are colorblind. Okay, we can do another one for practice. Carrier woman with normal vision. Men. Carrier woman. That means she does not have color blindness, but she has one colorblind gene. So that's XXC. We don't put C on both copies because she doesn't have colorblind. She's a carrier. She has a normal X to cover up the disease allele. Normal vision man is like this. Okay, I'll give you 2 minutes. Go figure it out, and then we'll take it out. All right, if anyone has an answer they want to share, I'm all ears. Okay, I got some answer coming in. So that's like that on the side, and then this like that on the top. And if we do it, so you can say 25% carrier female, which is like this one, right? And then 25% normal female, which are these ones. But remember, both of these are normal vision. Okay. And then what about boys? Okay, boys with 25% normal vision and 25% colorblind. You can say boy and girls, doesn't matter, okay? Do you understand? Okay. Sex-linked is the only thing I haven't quiz you on yet. So make sure you be careful with it, okay? All right, moving on. Just stop me, okay? Anytime. Oh, this is another one. Okay, I don't think I did this. Oh, actually, I did. I did this in class, but let's see if you remember. Okay, so we're looking at 6 genes here, right? And I want to know independent assortment, which means like whether you get big A, small a is not gonna influence on whether you get big B or small b, right? How you sort one gene is not going to affect other genes. That's basically what the independent assortment means. So over here, how many unique gametes? You might remember doing this question in class. Big A, small a, 2 choices. Big B, small b, 2 choices. Big C, 1 choice. Small d, 1 choice. 2 and 2. And in biology, if you have a bunch of numbers and you don't know what to do with it, what do you choose? You choose to multiply.
Multiplication.
There you go. Yeah, 2, 2, 2, 2, that's 16. Okay. Very simple math on the test, but if you want to bring a calculator to feel better, bring it. Bring it. Okay, give me a second here. Number 6 to 8. I don't know what is wrong with these numbers. There we go. A cat with long tail breeds with a cat with no tail. Long tail breeds with no tail. All the kittens have short tail. What inheritance pattern best describe this? Okay, let me do a poll with you. You know what? Never mind. Anybody want to answer before I tell you? Incomplete dominance. That is correct. Thank you. Thank you. Incomplete dominance. Because it's somewhere in the middle, right? It's like red and white flower gives you pink flower, somewhere in the middle. Okay. The heterozygous. Phenotype is an intermediate of the homozygous phenotypes. Okay, not long, not short, not long, not none, somewhere in between. Short. Brown eyes is dominant over blue eyes. Is it possible for 2 people with brown eyes to have a blue-eyed child? Like, this question always confuses people. 2 brown-eyed parents, can they have a blue-eyed kid? A for yes, B for no. What do you think? A for yes, B for no. Okay, we have overwhelming answer coming in saying yes. And Zoran had the answer in the chat box, actually. So if the parents are big B, small b, and you do the Punnett square, then there's a 25% chance of small b, right? Big B is brown, small b is blue, right? So if the parents are heterozygous, then there's a 25% chance. Okay. So is it always possible for brown-eyed parents to have a chance to have blue-eyed kids? Why not? The question is, Okay, yeah, you already answered it. Great. All right. If both parents are big B, all right, pure brown eyes, then all their kids are gonna be brown eyes, right? So you got to think about that. That's good. Okay, more sex-linked stuff. Janet has normal vision. That means she's going to be X, and then we're not really sure what the other one is. Her husband has normal vision. That means the husband must be X, Y. And we are asking, is it possible for them to have two sons where one has normal vision while the other is colorblind? Okay, well, let's take a look, okay? So obviously, if Janet is homozygous normal vision, then nobody's gonna have colorblind. Since the husband doesn't have colorblind, uh, if one of the kid has colorblind, it has to come from Janet, right? So we have to assume Janet is XXC, right? And then if we do the Punnett square, we will see that yes, right? If you only focus on the boys, if you only focus on the boys here in this question, then you will see, looking at boys alone, 50% chance the boy will be colorblind and 50% chance the boy will be normal vision. So yes, definitely. One son can have colorblind, the other doesn't. Is it possible for them to have two daughters where one has normal vision while the other is colorblind? Well, let's look at the daughter. We don't have to redo the Punnett square. We just have to look at the daughters. Okay, for daughters, it's either homozygous XX or heterozygous. But regardless, both of them have normal vision. Carrier is not colorblind. I have to emphasize that. So no. Okay. If it's a daughter, daughters are all normal. Okay. Normal as in like normal vision. Do you understand? Okay, you can see how I can challenge you on the test with these multiple choice questions, right? Which of the, Janet is normal vision, her husband is normal vision. Which of the following statement is true or false? Something like that, right? Okay. We're gonna do another question right here. This is number 9. Consider two traits: flower color and number of petals. Okay, so you should write it down on the test. Whatever the two traits are. Flower color. I'm not making sense here. Number of petals. So we have pure breed P generation, a pink flower with 6 petals, orange flowers, and 4 petals. So that means for the flower is either pink or orange. I don't know which one is dominant. And for number of petals, it is either 6 or 4. Okay, 6 or 4. But since they are pure breed, we can look at F1 to figure out which one is dominant. In F1, 100% is orange, and then with 6 petals, which means orange is dominant over pink, and 6 is dominant over 4. Okay. So, I mean, that's okay for now. Like you don't really have to do the letters necessarily. You can go and try to figure it out here. You cross two F1 individuals, so the F1s are all heterozygotes, which means this is the 9-3-3-1 situation. Okay? If there are 2,000 offspring, approximately how many will have orange flowers and 6 petals. Okay. So orange is a dominant trait and 6 petals, also dominant trait. So in the 9-3-3-1 situation, 9 is the dominant, right? So 9 out of 16 is going to be orange and with 6 petals. And we're talking about 2,000 offspring. So you multiply by 2,000, and that's going to give you the answer. Okay, 1125. Okay, you don't have to do the table. Okay, but you should explain your logic. Okay, like I verbally explained to you why I know orange is dominant and 6 is dominant, right? Okay, because these are the traits that shows up in the first generation, and the parents are pure breed. Okay, so that's how you know. Okay. Does anyone have any question about this one? And do you remember what these middle ones are, right? Can I explain again from the beginning or from like which portion? The 9331? Sure. Okay, so let me just type it out for you guys in case you need it, okay? If two parents, okay, so if both parents are homozygous for um, for both traits, okay? Then 100% of F1 are heterozygous for both genes, okay? Like in this case, color and petals. Okay? So basically, if you go F1 cross F1, then it's gonna be okay, which is gonna give you like a 4x4 table, right? So whenever you do the 4x4 table, the ratio is always going to be 9-3-3-1, like this, right here. This one. Okay. Remember we highlighted in class, right? So it doesn't matter what the two traits are. Okay, could be like brown and yellow, green, whatever, right? 9 out of 16 will always be the dominant trait and the dominant trait. The 3/16 would be one dominant trait and one recessive trait. And then 1/16 will be the recessive. So over here, because we are looking for orange and 6 petals, both are dominant trait. That's like the most common one. That's why we chose 9 over 16. 9 plus 3 plus 3 plus 1 is 16. If I say, orange with 4 petals, okay, because orange is a dominant trait and 4 petals is a recessive trait, then that's 3. 3 out of 16. And then I would times 2,000. Okay. If I'm looking for pink and 4 petals, then it will be 1 over 16 times 2,000. Is that a little bit better? So basically you have to know what the numbers represent, right? And then you choose the right number for your fractions, depending on which trait what trait combination you're looking for. Okay. You can only do 9-3-3-1 if you have the 16 4x4 table. Okay. On the quiz, like the question, you cannot use the 9-3-3-1 because on the quiz it was more like question like these. Okay, this is a 2x4 table. Then you cannot use 9321. You have to, physically count them. Okay. This is number 10. Okay, please pay attention to this one, okay? Consider the cross this and this. What is the probability of producing the genotype this? Now this is 3 traits. Okay, if we do the gametes, it would be like too many. Okay, don't like there are like 8 combinations here. 2 times 2 times 2 is 8. This one is 1 times 2 times 2, which is 4. If you do the table, 4 times 8 is 32. Okay, do not do table for this question. And so how do you do it? Well, it turns out you can do it separately, gene by gene. It's faster that way. So let's say we do just the A. Okay, so big A, small a, and then big A, big A. We're going to go ahead and do the table for it. Gonna do the table for it. Okay. By now, this should be automatic. Okay. There you go. And what's the question asking us to find? Which of these genotype are we looking for? Well, it says what's the probability of producing big A, right? So there's big A, big A. There's another big A right here. What fraction is that? One over two, right? Okay, go ahead and do it for B and do it for C. And remember, we're looking for different things. Okay, so go ahead, do the tables, and give me the fractions. Bring some highlighters. Highlight your answers. Make it easier for me to mark it, please. So, did you guys also get 1 over 2 here? Okay, and for the last one, also 1 over 2, I believe. Okay. So we have 3 fractions. And what did I always say? What do I always say? If you're not sure what to do with the numbers, what are we going to do? multiply. So that's 1/8. Okay. Now, for those of you who try to understand this mathematically, basically there's 50% chance of A being big A, and then 50% chance and 50% chance. If you have multiple things happening and you want them to happen at the same time, the overall chance of it happening together is the probability multiplied by each other. Okay, so that's half times half times half, which gives you 1/8. You need to understand this one, okay? Before I move on, any questions? Okay. If you change your mind, let me know. Okay, next. Hemophilia is an X-linked recessive bleeding disorder. Use the following information to create a pedigree. Okay, you should know this one as well, okay? I mean, you should know everything on the package for real, okay? But these ones, you absolutely must know. I wonder why. Okay, so hemophilia is X-linked recessive bleeding disorder, much like colorblindness. Use the following information to create a pedigree. Okay, so let's see. Alice has hemophilia and she marries Terry. Now, if you are confused, okay, like maybe do some background work first. Like I said, like there's a lot of time, okay? Don't rush it. Okay? A lot of you just hand it in like after half an hour. I don't know where you're trying to go to, okay? Just stay put, do it slowly, take your time. All right? So, do some groundwork first. Okay? Do some background stuff on the test before you dive right into the question, okay? So, first one here. This is normal. Normal men. Okay, so normal is XY. Normal woman. Okay, man, woman, female, male, whatever you want to say, okay? This is XX. This guy, hemophilia. Okay? Hemophiliac man. So what's his genotype? What is his genotype? Uh-oh, nobody knows. It doesn't matter what subscript you use, okay? You could keep with C if you want, or like you could use H for hemophiliac. But what is the genotype here? Okay, yeah. So we can go X, H, and then Y, right? Right? Or XC if you want, doesn't really matter. Okay, this is hemophiliac woman. What's the genotype? I'm gonna pick on someone, okay? Ruth, what's the answer? Sorry, it's just the first name I see in the class list there. What's the answer for this, Ruth?
XCX.
Uh, sorry, one more time. X what?
XH.
Uh-huh.
X. H.
There you go. Both of them has the H, right? Because she has the disease. Very good. Okay. And this one is normal woman, but she's a carrier. Okay, so what is her genotype? Benita, you want to try this one? You can do it. Oh, Angelina already has the answer there. Okay, sure. And Zoran, there we go. Right? Okay. We can put the H on the second one. So like I said, do some groundwork on the test. Write these down. It doesn't matter if it's hemophilia. It doesn't matter if it's color blindness or whatever X-linked recessive disease. Draw these things out. I'll even give you some marks. If you get the question wrong and you have these things on the side, I'll give you some marks. Okay. The more you write, the more opportunities I have to give you part marks, even though your answer is wrong, right? Okay. So yeah, show more work if you can. All right. Alice has hemophilia. Okay, so like we just Alice is a female, so we just look at our thing here and she has hemophilia. Okay, so it's this one. All right, so we're gonna put it right here. We're gonna go Alice. There's Alice. Okay, and you want to write Alice's name here. Okay, don't make me guess who it is. Okay, Alice, and she has hemophilia, so she's XHXH. Okay. Mary's Terry. Terry is normal. Okay, there's only one way to be normal for men. Okay, which is like just XY. Not there's no carrier situation going on. That's what I mean. And so this is Terry. Okay, so Alice and Terry have 2 daughters and a son. So how many shapes should I have here for the next generation? Sounds like a rhetorical question, but it's something to think about, right? How many shapes, huh? Hopefully in your head, you're thinking about 3. Okay, so 3. 2 daughters. 2 daughters and 1 son. Okay, so I'm gonna put 2 circles. I don't know what's going on with the daughters yet. We'll figure it out. There we go. One of the daughters married a man. Okay, good for her. Married a man. There you go. And they have a hemophiliac son and a hemophiliac daughter. So I had to go in other generation. Okay. What's going on with them? They have a hemophiliac son and a hemophiliac daughter. Okay, so there's the son, there's the daughter. On the test, I will give you names for these people as well. So you make sure you tell me who's who. Okay? Because some of you might decide to draw the square here and then circle, which is fine. Okay, but you gotta tell me who's who. All right, so let's see what's going on here. So this is hemophiliac son, hemophiliac daughter. So then you gotta think about what's going on with the previous generation. Well, where does this XH come from? Well, one of them has to come from the mother, and one of them has to come from the father. Right? I'll highlight it for you. This one is from mom, and this one is from dad. Right? So what's going on with dad? Dad must be hemophilia. Okay, because for men, if you have one disease X, you got the condition. Okay, and then what about the other one? Well, I'm not so sure. Let's take a look here. The XH has to come from the mother because the father doesn't have a disease X to pass on. In fact, the father can only pass on a normal X. Right? So that tells you this person must be a carrier. There's no other way. She's a carrier. Okay? And that's the family tree. Now, how about these people? Can we say anything about them? Well, the son is definitely colorblind. Sorry, that definitely has hemophilia because the dad can only pass on the Y, and the mom is definitely passing on a disease H. Okay. And what about the daughter? Do we know for sure what the daughter is, or we need more information? Do we know the daughter situation? Yeah, she's definitely a carrier, right? The father's gonna pass on a normal X from the father. That's all he can pass on to have a daughter. And the mother is gonna pass on a disease, a allele. So she's definitely a carrier. Okay, so that's the family tree. What is the probability that their next son also has hemophilia? Okay, so the question is asking about this family right here. And their next son will have hemophilia. Well, we can go ahead and do the Punnett square. That's the father. That's the mother. Oh my God, I can't draw a straight line. Okay, so I'm just gonna do the son here. Okay, so we're only asking if they're only looking at the son. We don't need to do the daughters. Okay, so 50% chance. All right. Practice drawing the pedigrees. Okay. Do you guys understand? You understand how to set it up with these symbols? All right.
Hi.
Hi,
Could
yeah.
you explain how do you create, like I did understand like these normal man is it's not color, but when you're creating the pedigree,
Mhm.
how do you create this one, the chart? I'm
So
so
sideways,
confused.
like, so do you understand this? This means getting married?
Yeah, that I kind of,
Yeah. So
yeah.
sideways, if you connect two people sideways, that means they're gonna have kids together. Okay. And then if you draw down, then it's gonna basically these are the kids, right? So based on the information that's given, that's how you can. Okay, maybe we do another one. In lecture 8, study guide. Okay, so this is another one. Okay. Give me a second. Okay, let's try again here. So a man and a woman both have normal vision. They have 3 children, all of whom marry people with normal vision. So let's start small. Okay, a man and a woman have normal vision. Okay, right here. Man and woman both have normal vision. So Manisha, what shape am I gonna use, huh? Since you were asking.
So both are normal reason. Sorry, what did you say?
A man and woman both have normal vision. So what shape should I draw?
It's like no color. Both of them are white.
Both of them are white, right? So,
X,
yeah. So what shape do I use for the man?
square,
Yeah. So there
and
is the man, and then
circle for the woman.
and should I connect them?
Yes,
Yes, because they're gonna have kids, right?
yes.
Now the man has normal vision. So what's his genotype?
XY.
XY. Okay. For the female, she has normal vision, which means she must have one normal X. But at this point, I don't know yet whether she is XX or XXC, right? She's probably XXC, because if she's not, then we have no question to do, right? But let's take a look. They have 3 children, all of whom marry people with normal vision. Okay, so three kids. We're gonna draw 3 kids. Okay, so first a son with color blindness. So that's gonna be the square. Okay, and then we're gonna shade it in. Right? That's the son. Okay, let's draw all the kids first. And then we're gonna have a daughter. We're not sure about what's going on with the daughter, but all her sons have normal vision. And then and then we're gonna have another daughter here. Okay. Now let's take a look at the son. Who's the son gonna marry? The son is gonna marry someone with normal vision. So he's gonna marry someone with normal vision. And the son is definitely XCY, which means the mother here is a carrier, right? And that's where the XC came from. Is this okay so far? For anyone? Like,
Yes, for me,
yeah.
yes.
Good. So what's going on with this? The son is gonna have a daughter with normal vision. Okay, so daughter, normal vision. Okay, so what does it mean? Well, it means the wife is, well, we already know the wife has normal vision. So she's X, and then we're not sure what it is. But for the daughter, she will definitely be getting an XC from the dad, and then the normal X from the mom, right? So like that. She's going to be a carrier, even though she has normal vision, right? The XC is from the dad. We have no information to figure out what the other genotype is, okay? So we just have to leave it like that. Now for the daughter, remember, the question says they all marry normal vision spouse. Okay, so this man is XY for sure. And 3 sons with normal vision. 3 sons with normal vision. Okay. All of them have normal vision. All of them are XY. Okay. So the woman must at least have 1 X. That's normal. Okay, and this normal X is the one that gets passed on to all the sons. Now we don't know if she's a carrier or not. Okay, we have no information about that. Right? We don't even know if this X came from the mother or from the father. Okay, it could be either one, right? Okay, so like we don't know. She could be a carrier, she might not be. Okay, and over here for the last one, the daughter's gonna marry a normal vision guy. Sorry, the shapes are going crazy. One second. Okay, normal vision guy. So he's going to be XY. And what happens is they're going to have two daughters. One, oh no, they're going to have what? 2 sons. One is colorblind and one is not. Okay, so colorblind son is XCY, normal son is XY. Remember, the Y came from the dad, which means the mother must be XXC. Okay, the Y is from the dad. Okay, so one of the XC is from mom. The other X is also from mom. Okay. In fact, this XC is from grandma, so to speak. Right? You see that? So that's how you do it. Now, on the test, if you see a pedigree question like this, there will be no question marks, okay? all the information in the question will tell you, will be enough to figure out everybody. In this particular example, we don't have enough information to figure out these two people. So that's why we have a question mark there. This would not be the case on the final test. You understand? Okay, so you must figure out everybody's makeup. Are we okay? Do you understand what I'm trying to tell you? Okay, this pedigree question is gonna be it's gonna be 5 marks, guys. Okay, I'm just gonna tell you whatever. 5 marks pedigree. So make sure you know how to draw. It's not gonna be more difficult than the ones we've done here. Okay, it's I'm gonna say it's somewhere between this one and this one. The level of difficulty. Okay? So you need everybody's genotype. You need to draw the connection properly, and then you got to figure out the shadings and everything, okay? Seriously, are we together? Okay. Next. Answer each of the following questions. How many autosomal chromosomes are found in normal human gametes? Okay, again, if you easily get confused about these things, like don't look at the questions yet. Just think about it. Okay? Normal cells 46, gametes 23 chromosomes, right? Okay. So here, how many autosomes in a normal gamete? Well, normal gamete has 23 chromosomes. If we don't count the sex chromosome, then that's 22. Right? 22. How many chromosomes are found in a normal human cell at prophase? Prophase, everything is still stuck together, so it's still 46. We don't count them differently. But in anaphase, the sister chromatids separate already, so everything is doubled. That's why it's 92. How many sex chromosomes are found in normal human zygote? Zygote is the thing you get after sperm plus egg. So it's already diploid. Okay, so in the diploid cell, 2 sex chromosome, XX or XY. How many sex chromosomes are found in normal human gametes? Normal human gametes is haploid, meaning it can only contain X or Y, so that's just 1. How many chromosomes are found in the cells of an individual with trisomy 18? Hey, someone help me with this one. Trisomy 18, how many chromosomes? What does trisomy 18 mean anyways? Yeah, trisomy 18 means extra chromosome 18. So normally people have 46. So now there's an extra one, 47. Here's another question that you must know. Okay. An organism has chromosome number 2n equals to 12. 2n equals to 12. And so let's remind ourselves what these number means. This is number of sets of chromosomes. This is number of chromosomes per set. And this is total number of chromosomes. Okay, The organism is haploid, diploid, or tetraploid. Well, they have 2 sets. 2 sets means diploid. How many sets of chromosomes does this somatic cell have? Well, I just told you 2 sets. Diploid means 2 sets. How many chromosomes does the cell have? 12. Once you know what the number means, it's easy. How many homologous chromosome pair does the organism have? Okay, 12 pairs. Right? Oh, sorry. My bad. I totally did this wrong. 6 chromosome per set, so 6 pair. Like chromosome 1, 1, 2, 2, 3, 3, 4, 4, ... 6, 6, right? So 6 pairs. 6 pairs. How many chromosomes does the gamete of this organism have? Gamete is always half of total. So total is 12, half of total is 6. Undergoing metaphase, everything is lined up. They have not separated yet. So I will have a bunch of these X's in the center of the cell. 12. Okay, let's try this question again. Let's do 3n equals to 36. Okay, 3n equals to 36. I'll give you a moment to fill this in. I'll be right back. Okay, so in this case, it would be, well, it's none of these answers, but 3 sets is triploid. How many sets of chromosome in this case? 3 sets, that's right. That's what the 3 means at the front. How many chromosomes does the cell have? 36. How many homologous chromosome pair? Well, it's not going to be a pair. It's going to be like triplets, right? Okay. Like obviously the question is going to correspond to it. So like 18, it's basically, 36 divided by 3, which is, no, not 18. 36 divided by 3 is 12. And where am I? How many chromosomes does it have in the gametes of this organism? Okay, so this is a trick question. Not possible. Okay, because it's odd set. If it's odd set, you cannot reproduce sexually. Remember, we talked about this in the watermelon case, a seedless watermelon, right? If it's odd set of chromosomes, they cannot make gamete. Okay. How many chromosomes does a cell undergoing metaphase have? 36. Okay. That's all. Okay, so I just want to repeat, odd set of chromosomes cannot do gamete formation. It's not the odd number of chromosomes. It's the odd number of set. Okay, you understand the difference, right? I hope. Okay, what else are we gonna do? We are going to do biotechnologies. Biotechnologies. There we go. What is the purpose of PCR? Okay, purpose of PCR is to clone DNA segment. Okay, make many copies of them. Make many copies of them. How does it work? I actually don't have, yeah, I don't have a question on how does it work, okay? So, yeah, never mind how does it work. But we talked about it in class, right? Like, with the primers and the tag polymerase. Do you have to know that? Let me see. Yeah, okay, this should be fine, okay? So if you start with 50 copies and you do 10 cycles, how many copies in the end? Do you remember the formula? I wrote it on the PowerPoint slides for you. How many copies? Not 100. How many copies? Okay, I guess it's good that we're reviewing this. Remember, is the initial number times 2 to the n, where n is the number of cycles. Okay, so in this case, initial is 50. That's what it says. Times 2 to the 10. 2 to the 10 is 1024. I guess you probably need a calculator for that. And then 50 times 1024 is, drum roll please, 51200. Okay, there you go. Just remember the formula. Okay, what are restriction enzymes? They cut DNA. I mean, these are relatively fresh stuff to you, right? We're still learning it last week, so I don't think they would give you too much trouble. But which one creates sticky ends? That's sticky end, that's sticky end, sticky ends, right? And then blunt ends are these ones. Okay, so which one are more useful? I mean, it depends on what you're trying to do. In class, we only talk about inserting a gene into a plasmid. Okay, we would need to create sticky ends because sticky ends allows the overhang, this thing that's hanging out, to reconnect with each other. Okay, and that would be ideal for putting things into the gene. We did number 3 already. We have a few other ones here that we should go through. Sorry, it looks like we're gonna go over time a little bit, but I'm glad we're doing this remotely. So hopefully you're at home. If you gotta go, just go. Okay, doesn't matter. Give examples of a palindromic sequence that's 6 nucleotides long. So if it's 6 nucleotides long, you just make up any 3. Okay, so like TAC. and then you copy it backwards on the other strand. And then you use complementary rules to figure out the rest. So T goes with A goes with T, G, and then you just copy it backwards again. Here's another example. CGA, write it backwards. And then just use complementary rule. So this way it reads the same on one way and then the same on the other strand backwards as well. Okay, 3 different DNA samples are treated with equal R1 and separated using gel electrophoresis. Remember, this is negative, this is positive, and the current go this way. What is the purpose of the lane of the marker lane? Okay, so this is called the ruler, right? For reference. Size reference. Okay, so like if I want to figure out how long is this one, then we can compare it with a ruler and it will give us the idea. So which sample has the longest DNA strand before being treated with equal R1? So over here, this one, I just read it off the ruler. It's 1,000. This one here is 800. Of course, it will be more clear on the test. This is 200. So 200 plus 800 is actually 1,000 again. 1,000. And then over here, 600, 300, and 100. And it also adds up to 1,000. So this is a trick question. Same size, same length before cutting. Which sample was not cut by the restriction enzyme? Right. A, right? Why? Only one band. So, oops. Okay. Explain what will happen if you make the following mistakes. Forget to load the dye, then sample will not sink into the well. Remember the video I showed you in class? The blue dye helps you makes it more dense and it helps the DNA go into the well. If you forget that, as soon as you add the DNA sample, it would maybe just shoot back up and you will lose the sample, okay? Sample loss. Reversed, sorry, spelling mistake. Reverse the positive and the negative terminal. So instead of current going down, the current's gonna go the other way. Remember, your sample is up here. So if the current goes the other way, the DNA is gonna run this way. And then it's gonna exit the gel very quickly. Okay? So samples move up the gel and exit. Sample loss. What if you leave the power on for too long? So the DNA will keep on moving. And when it gets to the end and the current is not turned off, it will keep moving and exit the gel at the bottom. Samples exit at the bottom. What happens then? Sample loss again. Okay. Like the longer one is probably going to be okay. Okay. Like if you look at the lane C, you probably will lose this one. Lane B, you will lose that one. Okay. But the other ones may be okay. Depends how long you left the power on, right? Forgot to add ethidium bromide, then DNA not visible under UV. Right? The ethidium bromide makes the DNA visible when you shine UV light at them. Gives us a way to see it. There are two DNA samples with STR polymorphism. This is the concept chart question we did in class. Just want to go through it again with you. This one is 3 repeats. Sorry, 3 nucleotides, 10 repeats. 3 times 10 is 30. This one is 3 times 15, which is 45. So which one's going to go down further in the gel? Well, the shorter one, right? The shorter one always goes further down the gel. Okay, next one. The plasmid below is treated with equal R1 when the digested plasmid is separated using gel electrophoresis. How many bands will you see? Okay, so equal R1 and BAMH. You cut here and you cut here. That's one band. That's another band. Okay. If I cut it with equal R1, Hindi 3, and small one cut, 2 cut, 3 cuts. That's 1, 2, 3 fragments. Now, one thing I do want to point out is if your DNA is linear, and then you do 3 cuts, you actually get additional piece, right? 4 pieces. Right, so read carefully on the test. See whether the sample is like plasmid to begin with or linear DNA. If it's plasmid, the cuts gives you the same number of pieces. Whereas here, you get an extra piece from the cuts. 3 cuts, 4 pieces. 2 cuts, 3 pieces. Okay, now there are these plasmid questions that we talked about, and then I posted like a worksheet online with practice, right, for you to do. So make sure you go through them, but I'll use this one as an example. Remember, this is antibiotic resistant gene to help distinguish, tell apart, okay? Help distinguish transformed bacteria and untransformed bacteria. Okay, transform means taken up the plasmid. Okay, untransformed, rejected the plasmid. If they have accepted the plasmid, they would also have resistance to whatever antibiotic is in the gene. Okay, as long as, and this is the important part, as long as when you insert the gene, you did not disrupt the resistant gene. So over here, you culture E. coli containing tetracycline. We insert using EcoR1. So EcoR1 is right here. The gene is going to go into this location. It will not disrupt the TETR gene. So you would expect some growth. Okay, some growth. Right. Why some growth and not like lots of growth? Because not every plasma, not every bacteria will take up the plasmid. Only some will do. So some growth. Okay, so you can elaborate. Only transformed bacteria can grow in the presence of tetracycline. Now, what if you insert using small one? You see, small one is right here, right? Small one. Then the gene's gonna go right there. Right? So insert,
no grow at all.
that's right. Inserting with small one will disrupt the TETR gene. What does that mean? Even if the bacteria accept the plasmid, it will not have tetracycline resistance. Nothing grows. Doesn't matter. Doesn't matter if they have been transformed or not. Nothing will grow. All right, I'll just give you a moment to copy that. Wait, you guys are 202, right? Oh my goodness, I think I just made a mistake here. Oh, no, 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 no. So, give me a second, guys. So on Luminate, the test 2 folder, there is the concept check answer. Okay, it's visible to you guys now. Okay, it wasn't visible before. Okay, what else do we have here? That's pretty much it. For the human body stuff, you can review yourself. Just know the superior, inferior, lateral, medial stuff. There are quite a few questions on those, but all of lecture 10 stuff will be in the multiple choice. 1, 2, 3, 4, 5, 6, 7, 7 questions from lecture 10. Okay, and all of it will be in the multiple choice section. Okay, so it should be pretty straightforward. Okay, let me stop the recording for a second here.